Hi, I'm Dominique Power.
And I'm Jaren Chang.
And we are from the University of Queensland. In vitro studies provide important insight into fundamental mechanisms for biological effects such as development or pathology. However, the caveat is always the fact that what occurs in an in vivo system might not necessarily reflect in vitro studies, particularly when you are looking at a heterogeneous population, such as human myotubes. Single-cell RNA-seq gives us a high-resolution overview of gene expression between cells. However, this can only be performed in whole single cells. Myotubes are derived from myoblast fusion. Therefore, single-cell RNA-seq cannot be utilized. Single nuclei RNA-seq provides us a new way to investigate this heterogeneous population to identify, in real time, gene expression. Our first aim was to isolate the single nuclei from human control, C19, C10 lines, and MND patients, M17, M20 myotubes, and secondary, to investigate if their gene profiles reflected biological variation in the context of adult myotube myogenesis. Myoblasts were extracted from human muscle biopsy tissue, then proliferated until desired confluency. After this time, they underwent myotube differentiation. At day 6, the myotube nuclei were isolated using a nuclear extraction method, stained for DAPI, and nuclei purified using FACS. The nuclei were then sent to a sequencing facility for preparation and establishment of the genomic libraries and RNA sequencing.
Once the data was finalized, a pre-processing pipeline was developed for this dataset. The results were visualized through 3D UMAP plots, which gives us a global overview of the variation of the transcriptional profiles. Through expert identification, we consistently found profiles that matched myotube expression, shown in maroon. Genes such as myo18B, TTN, and MYH3 DMD were found unique to this cluster. In the adjacent nuclei clusters, we identified genes involved in cytoskeletal rearrangement and extracellular matrix reorganization, which could identify myoblasts undergoing fusion. These marker genes suggest a linear set of events that could be congruent to adult myogenesis. Indeed, when we perform trajectory analysis on the nuclei, we see trajectories which potentially simulate adult myogenesis. Moreover, the heat map analysis enables a look at how gene expression changes through pseudotime. Genes with a high expression variability can be identified in the heat map, signified in yellow to red. Overall, this heat map parallels the marker gene analysis done for the clusters. We demonstrate snRNAseq is a viable tool that provides in-depth reads to profile and identify the heterogeneity observed in skeletal muscle. snRNAseq provides a new avenue to start identifying potential alterations in human MND myotubes versus control myotubes.
Thank you for your time.